Hello, welcome to Biotech Techno Solution Private Limited. We are going to discuss about the digestive system, and in particular we are going to discuss about the esophagus and the stomach. So these are the topics that we are going to cover in this session. We will be studying about the esophagus and the cardiac sphincter, that is playing an important role in the esophagus, followed by which we will be studying about the stomach, the functions, the sections, the lining of stomach, followed by the pyloric sphincter, your gastric glands, pepsinogen and pepsin. So these are the topics that we are going to cover in this session. Let us see what is esophagus. Esophagus is a straight muscular tube of 25 to 30 cm long, and it extends from pharynx to cardiac stomach. Your lower esophageal sphincter performs the function of preventing the stomach content from regurgitation. Regurgitation is nothing but the pushing off or throwing up of the food substances from the stomach back to the esophagus. So this process is prevented by your esophageal sphincter. And your esophageal sphincter also produces the heartburn. That is, the burning sensation that is produced by the entry of the acid into the esophagus. And these two situations are prevented by the esophageal sphincter. Your esophagus has the esophageal glands in submucosa, and your submucosa will secrete mucus. Your esophagus has skeletal muscle in the upper 1/3 and smooth muscle in the bottom 1/3. The middle 1/3 portion will be a mixture of the skeletal muscle and your smooth muscle. So here you can see your submucosal gland which produces the mucus, and your smooth muscle is over here. So your skeletal muscle is present in the esophagus in 1/3 upper portion and smooth muscle in the bottom 1/3. Next we are going to study about the cardiac sphincter. Your cardiac sphincter is nothing but a circular band of muscle which is located at the opening of the stomach. It controls the movement of the food into the stomach. So this is all about your cardiac sphincter. Next we are going to study in detail about the stomach. Your stomach is J-shaped, and it is a pouch-like organ which is about 25 to 30 centimeters long, and it hangs inferior to the diaphragm in the upper left portion of the abdominal cavity. The stomach has two layers of smooth muscle, which includes the inner circular layer which is formed of circular fibers, and the outer longitudinal layer which is formed using your longitudinal fibers. There may be a third layer which is made up of oblique fibers. So these are the various layers of the stomach. Let us see the functions of the stomach. The main function of the stomach is to receive the food from the esophagus and mix the food, that is the bolus, with the gastric juice. And once the bolus enters the stomach, it starts the protein digestion. And after the protein digestion, it is moved into your small intestine through the pyloric opening. Here you can see the pyloric opening. So these are the various sections of your stomach. You have the cardiac region, the fundus, the body, and the pylorus. So these are the various sections which are there in your stomach. Your food enters through your cardiac region, goes through the fundus and enters into the body of the stomach, followed by the protein digestion. It enters into the pylorus region, which has a pylorus canal. So this pyloric canal has a pyloric opening which opens into the duodenum, that is your small intestine's first part. Next we are going to see the linings of the stomach. So these are the following linings of the stomach. Rugae is the first lining that folds the inner lining, that is your gastric glands. Followed by rugae, you have the mucus cells, which are there to secrete the mucus and to protect the lining. Your chief cells will secrete pepsinogen, which converts into pepsin. And this pepsinogen that is converted into pepsin will help in protein digestion. So the protein digestion is done by the chief cells in major. Your parietal cells will produce the hydrochloric acid that is needed to convert your pepsinogen to pepsin. The intrinsic factor that is needed for the vitamin B12 for absorption is your pepsinogen to pepsin, and this is generated by the parietal cells. Next we are going to see about the pyloric sphincter. Your pyloric sphincter will control the movement of the substances into your small intestine. So here you can see that your pyloric sphincter has the control of the movement of substances from the small intestine to your duodenum. So here you can clearly see. You can see that you have the gastric glands. The gastric glands is stimulated by the parasympathetic nervous system. The gastric glands will secrete the hormone gastrin. And your small intestine secretes the hormone cholecystokinin, which inhibits the gastric glands. Your stomach can absorb the alcohol, water, and some fat-soluble drugs. In your gastric glands, you have the following layers. Your mucosa, your submucosa, your muscle layers, and the outer lining, that is your serosa. So this is how the gastric glands are present in a human stomach. The gastric pits are over here. So these gastric plates are formed by these pits. The mucous cells can be found in the magnified region over here. And the gastric glands, your parietal cell, your chief cell, is present in the mucosa layer. The term chyme is used for mentioning the mixture of food and gastric juices. So this forms your parasympathetic nervous system. Next we are going to see about the gastric secretions. So we studied about the mucus layer, which is containing the mucus cell, parietal cell, chief cell and G cell. So the mucous membrane of the stomach has gastric glands which secretes the following hormones, that is pepsinogen, pepsin and your hydrochloric acid. So these are the compounds that are secreted by the gastric glands. Your pepsinogen is generated by your chief cell, and they are nothing but the inactive form of pepsin. Your pepsin, which is produced from the pepsinogen, is done in the presence of hydrochloric acid. Your pepsin is nothing but a protein splitting enzyme. So that is about the pepsin. Next we have the hydrochloric acid. Your parietal cells, as you can see over here, is the one that secretes the hydrochloric acid. And the hydrochloric acid is used for converting your pepsinogen into pepsin. So your parietal chief cells form the major cells, which helps in the production of pepsinogen and hydrochloric acid, which produces the pepsin as a byproduct. Next we are going to see the regulation of the gastric secretions. Here you can see that your parasympathetic preganglionic nerve fiber over here, after which your parasympathetic postganglionic impulses will stimulate the release of gastric juice from your gastric glands, followed by which the impulses will stimulate the release of the gastrin, and after which you have the gastrin, which stimulates the gastric glands to release more gastric juice from the bloodstream. So this is how the gastric secretions occur. So here you can see how the mixing and the emptying actions are done in your stomach. So initially you can see that your food is entering over here, and after which your pyloric sphincter is contracted in order to safeguard your food from moving into your duodenum. Once the food is mixed with the gastric juices, then your chyme is produced, which is a mixture of the food and gastric juices. After which your pyloric sphincter is relaxed and your food enters your duodenum. So this is how the stomach performs the production of chyme, that is the initial stage of your digestion of proteins. Next we have the mixing and the emptying actions. First, your duodenum fills with the chyme, and after which the sensory stretch receptors are stimulated. So once your sensory receptors are stimulated, you have the sensory nerve impulses to travel to your central nervous system through your vagus nerve. And after your central nervous system receives the impulse, it travels to inhibit the peristalsis in stomach wall. So by which the chyme that is present in your stomach moves into your duodenum and empties the stomach. Next we are going to see about the small intestine. The small intestine is a tubular organ that extends from the pyloric sphincter to the beginning of the large intestine. It completes the digestion of the nutrients in chyme, and it absorbs the products of digestion, and it transports the remaining residue to the large intestine. So this is how the small intestine helps in digestion. It consists of three parts. Your small intestine has three parts, which are nothing but the duodenum, the jejunum, and the ileum. Here you can see it in highlighted colors. So these parts form your small intestine, and it helps in the completion of the digestion by absorbing the products of the chyme. So this is how your small intestine helps in the digestion part. Next we are going to see about the part mesentery in your small intestine. Mesentery is nothing but a fan-like tissue which holds the jejunum and the ileum in the abdominal cavity. It attaches to the posterior wall of the abdomen, which is your ileocecal sphincter. It controls the movement of the chyme, that is, the mixture of food and gastric juice from ileum to the cecum of your large intestine. So this is how your small intestine is bound together in a particular shape with the fan-like tissue known as your mesentery. Here you can see how the mesentery will look like. So this is how the mesentery looks with your small intestine. It is a tissue that binds together the small intestine, which is extending from the stomach through the duodenum, jejunum and ileum into the large intestine's cecum. Next, we are going to see about the movements of the small intestine. Your small intestine carries on mixing movements which include the peristalsis, that is the pushing movements that propel the chyme. And next we have the segmentation, which is a ring-like contraction that can move chyme back and forth. So this is how the small intestine helps in digestion and movement. So here you can see that peristaltic is nothing but the alternate contraction and expansion which moves the chyme inside your small intestine. Segmentation is ring-like contraction which moves the chyme back and forth. Let us study about the secretions of small intestine. In addition to your mucus secreting goblet cells, there are many specialized mucus secreting glands, which are the Brunner's glands, that secrete a thick alkaline mucus in response to that of a certain stimuli. So your Brunner's cells or the glands secretes your Brunner's glands secrete the mucus and certain enzymes which helps in digestion. Your enzymes in the membranes of your microvilli include the peptidase, which break down the peptides into amino acids, followed by which you have the sucrase, maltase and lactase enzymes, which breaks down your disaccharides into monosaccharides. Next we have the enzyme lipase, which breaks down the fats into fatty acid and glycerol. Our next enzyme is enterokinase, which converts your trypsinogen to trypsin. And next we have the somatostatin, which is a hormone that inhibits the acid secretion by the stomach. So this is how your digestion is handled by the enzymes which are secreted in your microvilli. And the enzymes are like peptidase, sucrase, maltase, lactase, lipase, enterokinase and somatostatin. Next we are going to see about the regulation of small intestinal secretions. The regulation of small intestinal secretion occurs by the mucus secretion, which is stimulated by the presence of chyme which enters into your small intestine. The distension of the intestinal wall activates the nerve plexus in the wall of the small intestine. The parasympathetic reflexes triggers the release of the intestinal enzymes. So this is which stimulates the small intestine to produce the secretions of the enzymes which helps in the digestion, converting your proteins into simpler compounds which can be easily absorbed by your body. Next, we are going to see the internal structure of your intestine. On enlarging your intestine, you can find that it has a sort of finger-like structures. The finger-like structures are known as the villi. The villi are the structures which helps in absorptions, and they are known as, and the total protrusion over here is known as placae circularis. Your intestine has the submucosa to be the internal lining, followed by the circular muscle, above which you have your longitudinal muscle and serosa over here. So these are the muscular layer of the small intestine, and which is a smooth muscle, as you can see. Next, we are going to see the walls of the small intestine in detail. So if you see, your small intestine has the microvillus or the microvilli, which are finger-like structures, and they are protruding on the walls of your small intestine. Next, we have the cell membrane, and in your cell you have the mitochondrion, the Golgi apparatus, your nucleolus, which is present inside the nucleus, and the rough endoplasmic reticulum. So these are the components of the walls in your small intestine. Next, we are going to see about the structure of the villi, which are finger-like projections which you can find in your small intestine. Villi is a plural form, and for a singular one we can say that as villus. So your villus will have a simple columnar epithelium, inside which you will be having the blood capillary network, and your blood capillary network encapsulates the lacteal. In your epithelium you can find the goblet cells. Over here you can see your goblet cells. And your capillaries has the arteriole, the venule and lymph vessel, which is present in your villus. So this shows the structure of the villus. Next, we are going to see how the absorption is done in your small intestine. The villi will increase the surface area for absorption. Your small intestine's absorption is so effective that very little reaches the organ's distal end, that is, to the end you don't have anything left for absorption. So the most of the things which a person's stomach and the small intestine can absorb is done effectively with the increase in the surface area of the villi. Your small intestine has the monosaccharides and the amino acids, which is absorbed by your small intestine through the villus. And how does villus absorb? That is done by diffusion and active transport. The nutrients that you can find in your food, that is occurring at the stage right now, will be absorbed into your blood through the villus. The large proteins are broken down and they are absorbed into your villi, which in turn will be transferred through your blood to the various parts of your body. Your fatty acids and glycerol absorb by several steps involved and absorbed into your lymph and blood. So your fatty acids and glycerol that is present in your food is absorbed into your lymph and then into your blood. So this is how the absorption takes place in your small intestine. Your monosaccharides and amino acids through diffusion enter into your blood and is transported to the various parts of your body. Next we are going to see the molecular reactions. So the food that enters into your stomach is known as a chyme, and the chyme has the food structure, that is your maltose disaccharide, as this. Your maltose disaccharide, on addition with the water in the presence of maltase, will produce glucose, that is energy. So your disaccharide is converted into a monosaccharide, as you can see. Your H2O, here you have 2 hydrogen molecule and 1 oxygen. So this is added over here. You can see that one of the hydrogen is added to your benzene structure over here, and your remaining oxygen and hydrogen is added to the benzene structure over here. So this is how your maltose disaccharide in the presence of water and in the presence of maltase is converted into glucose. Whereas your dipeptide from your protein digestion in the presence of dipeptidase, that is your water, it produces amino acid. So your dipeptide with water in the presence of dipeptidase produces the amino acids. Your 2 hydrogen and 1 oxygen is added over here in this region, as you can see, as OH and H. So this is your dipeptide from protein digestion with water in the presence of dipeptidase will produce amino acids. So this is how you can explain the process of digestion in chemical equation method. The food that enters with water will produce glucose that produces energy. Your dipeptide in the presence of water will produce amino acids, that is, breaking down of your protein complex into simple amino acids which can be easily digested and absorbed by a human body. So we are breaking down the protein structure, dipeptide, into amino acid by replacing the bonds over here with OH and H. Next we are going to see the molecular reaction for fat. So this is the structure, molecular structure of fat. With water in the presence of lipase, it gets converted into fatty acids and glycerol. So your fat has the structure over here like this. And as you can see, this portion, on addition with the hydrogen, can be glycerol. So here, this is how the structure is split into C17H35 C, single bond C. Your oxygen goes over here in double bond, and here you have 6 hydrogen and 3 oxygen. So your 3 oxygen and hydrogens are joined over here in your fatty acid, and your hydrogen goes over here, making this compound into a glycerol. So this is how the molecular reaction takes place in a human's digestion system, and how the fat will be converted into fatty acids and glycerol with water in the presence of lipase enzyme. So this is the molecular reaction, how the fat is being digested into simple compounds which can be easily absorbed by the body. Thank you so much for joining GTech on digestive systems of human system. Hope you would have learned lot of things about the esophagus and stomach. Thank you.